This lesson's about linked genes, and here are your objectives. Mendel did his work on heritable factors, what we would now call genes, in the mid-1800s. And although his work was initially forgotten, it was rediscovered in the early 1900s. And also, between Mendel's time and the early 1900s, the details of mitosis and meiosis and how chromosomes were involved in those started to get worked out. So these two lines of thinking, genes and chromosomes, started to come together, and scientists began to ask, are genes on chromosomes? And there seemed to be some good reasons for thinking that they might be. Both genes and chromosomes were known to be paired in diploid cells. They both separated in meiosis, and they both paired back up in fertilization. So it made sense that genes might be on chromosomes, but there wasn't strong evidence for it yet. Evidence that genes were on chromosomes came with the discovery of linked genes. So to understand linked genes, let's take a look at one of Mendel's dihybrid crosses that we've looked at in a previous lesson. So this is a cross between two heterozygotes at two different loci, like this. And the ratio that you end up with is a 9:3 to 3:1 ratio. And the observation that Mendel made and the conclusion that he drew is that all combinations of alleles in the gametes are equally likely. And he formulated that into his law of independent assortment. The alleles of one gene assort independently of the alleles from another gene. The 9:3 to 3:1 ratio that you see here is really two 3:1 ratios superimposed on each other, showing that the alleles of each gene is assorting independently from the other. But what if he had gotten different results instead? Let's imagine a test cross where we take a heterozygote like we had above and we cross it with a homozygous recessive. Remember from our previous lessons that yellow is dominant to green and round is dominant to wrinkled, so this would be yellow and round, and this other parent would be green and wrinkled in phenotype. The heterozygote can produce these four gametes: dominant dominant, dominant recessive, recessive dominant, and recessive recessive. And from the test cross homozygous recessive parent, we can just get the two recessive alleles. If we put those together, we get four possible combinations of our alleles. And we get four possible different phenotypes. We have yellow and round, which is one of the same phenotypes as the parent. We have green and wrinkled, also like one of the parents. But then we also have green and round, and yellow and wrinkled. And so we would call the ones that look like the parent, we would call these offspring the parental phenotype. And the others that don't look like either parent, we would call recombinants. They have the recombinant phenotype. And if the alleles assort independently, then we would expect a ratio of 1:1 to 1:1 in this test cross. And we would say that the genes are unlinked. They are assorting independently. Well, what if the hypothesis that genes are on chromosomes is correct? And what if the Y and R genes are on the same chromosome? Let's imagine here the two chromosomes from this parent. And I'm going to put the dominant alleles on the same chromosome. There's no reason why I have to do that. I could have put them separately, but I'm going to put them on the same chromosome for the point of this outline. The recessive homozygote just has the recessive alleles on both chromosomes. So if we could look at their chromosomes, it would look like this. Now that changes things, because if we assume that the chromosomes remain intact in meiosis, then one chromosome could be passed to the gamete or the other. In that case, there are really only two possibilities here in terms of the gametes from the heterozygote parent. They could pass along dominant-dominant, or they could pass along recessive-recessive. They can't pass along this one or this one because those are not how the alleles are arranged on their chromosomes. In that case, if the genes are on the same chromosome, if they are completely linked here, we would expect to see a 1:1 ratio of the yellow round to green wrinkled, and we'd expect to see none of these recombinants. And observing a ratio like this one instead of like this one would give us evidence that genes were on chromosomes, because it's chromosomes that are providing the physical structure that is linking the Y and R genes together. And in the early 1900s, American geneticist Thomas Hunt Morgan discovered linked genes, genes that are inherited together, when he was crossing Drosophila melanogaster, or what we know of as the fruit fly. And more than just discovering genes that were linked to each other, he was able to show that some genes were inherited in the way that chromosomes were known to be passed along. Drosophila has sex chromosomes like humans do, XX for female, XY for male, and he was able to show that the gene for eye color was inherited in the same way as the X chromosome. We would say that eye color is sex-linked. It's passed along differently depending on whether the offspring is male or female. So taken together, this was evidence that genes are on chromosomes. Now let's just back up to here to think about some of the results that Morgan obtained. Again, this is a hypothetical experiment. It turns out that in Mendel's experiments, all of the genes were unlinked. So the genes for P color and P shape are unlinked. So we do end up with the expected ratio here in a test cross of 1 to 1 to 1 to 1. But let's just use this, continue to use this to illustrate something. So instead of just having the completely linked situation like we have here with the 1 to 0 to 0 to 1 ratio, Morgan observed things like this, a 1 to 0.2 to 0.2 to 1 ratio, where we have much fewer of the recombinants than the parental phenotypes, but we still have some. So what's going on there? Well, let's think about what meiosis 1 would look like in the case where the genes are on different chromosomes or if they're on the same chromosome. Here I'm using red and blue to indicate the maternal and paternal homologs, and I'm going to draw long and short to indicate two different chromosomes. So if the Y and R genes are on two different chromosomes, that might be my dominant Y, here's my recessive Y, and here's my dominant R, and here's my recessive R. And because of independent assortment in meiosis 1, you can get any combination of the Y and R gene as these move to opposite poles. In contrast, if we have both genes on the same chromosome, like this, where I'm going to again draw the dominant ones on the same chromosome. They don't have to be, but that's just what I did in my example before. And I'm going to put the recessive ones here and here. Now, if they are on the same chromosome, as these move to opposite poles in meiosis 1, I'm going to get the dominant of both or the recessive of both most of the time. But remember that crossing over happens in meiosis 1, and so I could have crossing over between these two non-sister chromatids. And that could recombine the alleles into new configurations. So here I have the recessive versions of both the Y and R genes, and here I have the dominant versions of both the Y and R genes in their original configuration on the chromosomes. But if I have crossing over here, then I could end up with a new chromosome that has the recessive Y allele and the dominant R allele. And I could end up with a new chromosome that has the dominant Y allele and the recessive R allele. So it's crossing over in meiosis 1 that can lead to those recombinant gametes that give us the recombinant phenotypes. So that's where these phenotypes came from in our hypothetical example, because now I can end up with a gamete that has the dominant Y allele and the recessive R allele, and also one with the recessive Y allele and the dominant R, by crossing over between the Y and the R genes. Notice also that these are less frequent than the parental phenotypes because they require a crossing over event, and also that their frequency is the same because the crossing over event that produces one also produces the other. We would say that the Y and R genes here are linked because they're located on the same chromosome and they tend to get inherited together, but they can be separated by crossing over, and the frequency with which they would be separated depends on how close they are together. Just one more reminder that this example from peas is made up. In Mendel's actual pea experiments, the genes for pea color and pea shape actually are completely unlinked. They're on separate chromosomes. So in summary, linked genes are inherited together. That is, they do not assort independently. And that happens because they are located on the same chromosome, and they're also close together. If they're far enough apart, crossing over will happen frequently enough that it'll look in the results of the crosses as if they're on separate chromosomes. Now, by observing the frequency of the recombinant phenotypes, what we saw there as 0.2 in our hypothetical example, that would actually allow us to map out a chromosome, because the closer two genes are on a chromosome, the less likely their alleles are going to be separated by crossing over. So here, if the A and B genes are close together and the C gene is far apart, we'll see recombinants of A and C and B and C quite frequently, but A and B will still tend to be inherited together. And so by doing lots and lots of crosses and observing the frequency of the different recombinants, we could actually map out the chromosome. So that's an introduction to linked genes.